Okay, so let's go ahead and review what we talked about yesterday with respect to RNA. Here are the 4 characters in our story. Careless character 1, 2, and 3. All 3 characters, what do they have in common, people? What do they have in common?
They're all RNAs.
Very good. They're all RNA molecules, okay? So RNA macromolecules more correctly, and so what does that mean? That means that they're all made out of nucleotides, right? Remember a nucleotide looks like this, okay? And it has a nitrogenous base here, okay? And the biggest difference was their little nucleotides just differ by this, like it has a little extra, it has an extra oxygen. And then remember, instead of a thymine, it doesn't have thymine anymore. Okay, there's nucleotides that have uracil. So anyways, so all these guys are made out of RNA nucleotides, and that's why they're RNA molecules. And they're also, if you notice, they're single-stranded. It's just a single strand, all right? Okay, the first one, what is the first RNA molecule called?
The messenger.
Good, it's called mRNA, very good. mRNA, messenger RNA, very good. Number 2, what's number 2 called?
tRNA.
A tRNA, very good. A tRNA, excellent job. And the next one, what's the next one called?
Ribosomes.
Ribosomes, very good. This one's a ribosome. And ribosomes are a little weird because they have, they're made out of RNA, ribosomal RNA, that's what it's called. Okay, and the rest of the molecule, the rest of the ribosome is made out of protein. So, but ribosomes, as we know, why are they super important? Anybody? Why are ribosomes super important?
They make protein.
Very good. They're protein factories. Very good. Protein factories. That's what they're using in the book now. Factories. Excellent job. All right, they're protein factories. Now, here's the next one. Here's the next one. All right. The next one, okay, so now all of these RNA molecules are made where? Where are these RNA molecules made or constructed? Where?
No. In the cell.
In the cell, but where in the cell?
The nucleus.
The nucleus.
That's right, the nucleus. And here we have a nucleus, right? And remember, every single RNA molecule is a copy of DNA. It's a copy of a segment of DNA. Okay, it's a piece, a copy of a piece of DNA. So for example, from here to here, let's call that a piece of DNA, right? And what RNA polymerase does, it copies that piece. Okay, got it? And when that piece has instructions for making proteins, we call that an mRNA molecule. So let's just imagine that this right here, that's our mRNA molecule. Okay, got it? So far so good. We know that the mRNA is made in the nucleus. Okay, we know it's made by this special enzyme called RNA polymerase. Now, who can tell me what's going to happen to this mRNA before it leaves the nucleus? What has to happen to it? What has to happen to this mRNA before it leaves the nucleus? We talked about it yesterday. Someone has it. I know you guys do. You do. What was that, Santana?
It needs to be edited.
That's right. It needs to be edited. Very good. Edited. So this mRNA, right? This mRNA, pretend is this thing here, right? And so it has to be edited, and then so we can have a final version. All right. And so that's what happens. And so in the book, it talked a lot about how this might be a like an evolutionary like thing that helps us make lots of different proteins from one piece of RNA. So that's something to think about. Okay, so yes, it has to be edited. Now, who remembers the 2 words that are important and when we're talking about editing mRNA? What were the 2 terms that were super important? One started with an E and one started with an I.
Extrons and introns.
Very good. Exons and introns. Very good. Exactly. So there you go. Exons and introns, right? And so let's take a look at what that looks like, okay? So now here we have an mRNA molecule. Okay? There's an mRNA molecule, right? Let's imagine that this piece right here is an exon. Let's imagine that this piece here is an intron. And I'm just doing this very arbitrarily, okay? I'm just picking random things, okay? And then this part, let's imagine that's an exon, okay? Now, what happens to introns?
They get cut out.
Very good. They get cut out. Very good, right? And so now think about it right now. How many codons are in this mRNA right now before I edit it? How many codons?
2.
4.
4. I have 2, 4. Any other answers? How many codons?
3.
3. Okay, so the correct answer is 1, 2, 3. There's 3 codons. Remember what a codon is. It's just 3 consecutive nucleotides. Does that make sense? 3 consecutive nucleotides. And so this one is not a codon because there's not a third one, right? And so look how editing changes things, okay? Got it? Right now, if I was making this protein, the AUG, what amino acid would that tell the ribosome to start with? What amino acid? Because remember, these are just instructions for the protein factory, right? So like, here comes the instructions. Oh, the order's in. Okay, we have to make this protein. Okay, what protein? How's it gonna start making the protein? What's the first amino acid it's gonna put together? A, U. Whoops. Well, my bad. A, U. Right. We have to stay in this quadrant here. AUG, what's that right here? I know it's hard to see for you guys, but that is methionine. UAC, UAC, that's tyrosine. And the next one is UCG. UCG, that's serine. Right? So this would be the protein right here if we were using this RNA this mRNA by how it looks, right? But remember, we said that CU is an intron. So check this out. Look, I'm going to cut out the intron, right? See that? And so now these 2 exons get spliced together. You see that? Now, look at my codons now. My codons, 1, 2, 3. I still have 3 codons, but AUG, that's going to give me still methionine. UAC, UAC, that's still tyrosine. But now look, the last codon, GUA. GUA, that's valine. See that? And so after the mRNA got edited, right? Okay, the intron got cut out. Now look at the protein that we made. Is it the same as the one on the top?
No.
No, that's right. It's not the same. And remember, what a protein's, write this down, guys. A protein's abilities, okay, are based on their sequence. of amino acids? Because remember what we said, guys. We said that proteins are like apps, right? And so they have specific jobs that they have to do. And it's the amino acid sequence, the order in which the amino acids are connected, that determines the ability of the protein to do its job. So for example, a protein with methionine, tyrosine, and serine, okay, that would do let's call it job A. But this protein, because it has a different amino acid sequence, that would do a different job. Okay, let's call that job B, right? And so that's how proteins are different, because they have different sequence of amino acids and therefore they do different jobs. Okay, questions on this, guys? Questions, concerns on this? So far, okay, you guys should be really good with respect to this. So if we're talking about our learning goals, all right, you guys should all be able to do this now. You should, if I give you a piece of DNA, you should be able to give me what the mRNA looks like. If I give you a piece of RNA, like I just did, you should be able to tell me what the protein looks like. And then also you should know the difference between exons and introns. Okay? Right? And if you guys, raise your hand if you feel good about the first one. Raise your hand if you feel good about the first one. Good. How about the second one? Good, good. And then the last one, do you know the difference between an intron and an exon? Right? Okay. So if you guys understand these three things, okay, you're in a really good spot. The last thing I want to show you is this video here of what we've been talking about. And this video is in your agenda here. Okay. Your agenda. Okay, notice here it is. Let me zoom this. Let me speed it up a little bit.
Here is a cell, the basic unit of all living tissue.
All right, so remember, okay, we're talking about what's happening in the nucleus first. Elizabeth, what happens in the nucleus?
It makes DNA and RNA.
Yes, and then in the nucleus, you do DNA replication. But right now, we've been talking about which one? Transcription or translation? What happens in the nucleus?
Transcription.
Good. And so now there's the piece of DNA that we want to copy, right? Okay, here comes RNA polymerase, opens up the DNA, and here comes the RNA nucleotides. And see how they're being stringed together to form the messenger RNA? That's what's happening. Okay, and then once it's done, the messenger RNA now has to be, what's going to happen to this guy? It has to be what? Starts with an E.
Edited.
Edited, exactly, edited. Very good. This thing has to be edited. So the intron, all right, has to be cut out, right? And so that's what you're going to see in the video, right? There's going to be a piece that's going to be cut out, and then the exons get spliced together, and then that final mRNA goes out of the nucleus to a ribosome, and the ribosome is going to grab onto it. And now we have our protein factory, right, that we talked about. We got our messenger RNA, the instructions, and now this protein factory is going to produce the protein that we need by following these instructions that got sent from the ribosome. And this is the part that we're going to talk about next time, okay? And so make sure that you guys kind of digest this. Any questions with respect to today's material? Any questions, concerns? All right, good job.